The overall goal of this procedure is to perform ligating a PCR product into a plasmid having topoisomerase coupled to distinguish the main elements containing a vector with its function by selecting clones by blue or white coloring.
Topoisomerase cloning was born due to the need to link any PCR product with a plasmid without having to add restriction sites for encouraging restriction enzymes activities. First of all, topoisomerase makes a break in one of the DNA chains to uncoil and release tension by remaining bound to the DNA. Then it reattaches DNA fragments that are needed from each other. Thus, this topoisomerase function has been exploited to carry out cloning systems for PCR products. Topoisomerases bind to a 5' CCCTT specific sequence upon a DNA fragment plasmid and break a phosphodiester skeleton in one only change, bringing about an impaired timing. Topoisomerases remain bound covalently to the vector through an interaction between its tyrosine 274 and a phosphate group in the cleavage site. Furthermore, this bond becomes destabilized during the presence of a PCR product that contains an adenine in both extremes, coming along with topoisomerase releases. According to Thermo Fisher Scientific Catalog, PCR 2.1 topo is well recognized as a vector for the direct ligation of TAC-amplified PCR products, yielding up to 95% recombinance. Thus, it is widely used for the T8 topoisomerase cloning system. There are some features that distinguish PCR 2.1 topo among other vectors such as T7 promoter and M13 forward and reverse primer sites for in vitro RNA transcription and sequencing. Equerry sites flanking the PCR product, insertion site for easy excision of inserts, easy blue-white colony screening for selection of recombinants, kanamycin and ampicillin resistance for one's choice of selection in E. coli. Salt concentration will depend on the type of transformation that will be employed, chemical transformation and electroporation. For instance, if it's a chemical transformation, a saline solution will be used just as the provider, Thermo Fisher Scientific, points it out. In this case, for a reaction of 6 microliters, it is preferable to use 1 microliter of a saline solution due to the duty of neutralizing electric charges in cell's membrane, whereas for an electroporation, a saline solution will have to be diluted in a 1 with respect 4 ratio, accounting for 1 microliter of the total reaction volume, due to the need to have a cell suspension with a high resistance and low conductivity before receiving an electric pulse in an electroporator. PCR products must have sticky ends or cohesive ends. In other words, a nitrogenous base ought to be left without being paired at 3' extremes of the direct and inverse sequences. It is usually added an adenine into 3' extremes.
xGal is an indicator for cells expressing beta-galactosidase enzyme encoded in LAC-Z genes of the LAC operon. xGal is hydrolyzed by beta-galactosidase and galactose 5-bromochloro-3 hydroxyindole. The latter is oxidized to 5,5-D-bromo-4,4-D-chloro-indigo blue insoluble compound. Thus, if xGal and an inducer of beta-galactosidase are dissolved, IPTG, in the agar medium of a culture plate, the colonies grown on the plate having a LAC-Z functional gene can be clearly distinguished by its blue color. In addition to its characteristic color, volatilization indole group produces a characteristic smell bad. When cloning a gene into a plasmid vector is being done in a laboratory, xGal is used in this way to visualize the transformed bacteria colonies that have not cut the vector with the gene. Usually, they use this for purpose cells of the bacterium E. coli, which cannot produce the beta-galactosidase enzyme. Once transformed, the bacteria carrying the plasmid are not able to produce the enzyme beta-galactosidase because the region that synthesizes the enzyme part to make way for the recombinant sequence. Bacteria that do not have the recombinant sequence can break the xGal present in the agar medium colonies becoming blue. Bacteria that have been transformed, they cannot process xGal and they remain with their natural white color. But also to verify that the obtained colonies have the desired insert, a digestion with restriction enzymes can be done, and verify the digestion with an electrophoresis gel. Also, this can be done with a medium containing an antibiotic when the plasmid desired to insert contains an antibiotic resistance gene. There also exists the possibility of using flags or tags or fusion proteins, protease cleavage sites, etc.
The following protocol for ligation of a PCR product in the PCR TM 2.1 topo vector by complementarity in extreme TA timing adenine is based on the recommendations plasmid. PCR product thawed on ice. PCR 2.1 topo plasmid and the kit reagents are thawed and kept on ice. The ligation reaction for chemical transformation is presented at the following table. Incubate at room temperature for 10 minutes. 3 microliters ligation in 50 milliliters of chemically competent cells were transformed, using cells that had given you better transformation efficiency in the prior practice.
After transforming, 300 microliters of sock medium were added. The solution was incubated at 37 degrees. For recovery of the bacteria, it should be stirred for 1 hour while incubating. Entire volume of an LV plate with ampicillin 100 grams per milliliter and X-cal should be spread with a spatula. Leave to 37 degrees, incubating the plate overnight. Once the results were registered, the plate must be kept in the refrigerator.
There was no growth in the petri dish. However, there was used a plate from another team where colonies grew successfully. In Figure 1, on the left image can be seen cells that were transformed, represented by blue and white colonies shown as the dots. The right image shows the total UFC counting through paint. From the 71 colonies, only 3 were colored blue and 68 were white. The efficiency was calculated as shown in the screen.
PCR is a worldwide genetics engineering technique to amplify genes or sequences that would like to be cloned in vectors for the expression of recombinant proteins, for generating mutants, and for sequenciation or characterizations. Inserting these fragments of interest in a vector requires that exists a complementary sequence among them in order to ligate them with the vector. One of the techniques applied for having these complementary sequences is by means of restriction enzymes. However, not always restriction sites belonging to the multiple cloning sites and the insert of interest will coincide. Thus, there is a TA topoisomerase cloning system that uses a vector called PCR 2.1 topo in order to ligate any PCR product without the need to use restriction enzymes. According to the provider Thermo Fisher Scientific in vitro gene life technologies, there are key elements in a PCR 2.1 topo vector: lag set alpha fragment, an M13 reverse and forward priming sites, multiple cloning site, T7 promoter, an F1 origin, kanamycin and ampicillin resistance genes. There are other variants that take into account topo vectors, such as blunt ends cloning, directional cloning. There are several ways to identify or to verify if a ligation has been perfectly made. Gel electrophoresis, by separating, identifying, and purifying the linearized plasmid, the supercoiled plasmid, the plasmid without the insert of interest, and the plasmid plus the insert of interest. We have a second one that is antibiotic resistance. This is based on growing bacteria on LB plates with some antibiotics. Then we have the blue and white colored colonies screening. An example could be the alpha complementation that was previously explained.
Selecting clones by blue-white coloring is not 100% trustable. This method lets us to know which colonies are modified, but it can't tell if the gene was inserted in the correct direction. But if we use variants of topo vector family, we can then know if they were ligated directionally, because of the blunt ends that can be selected of one side, not from the other one. Also, if the lecture frame is not respected, then we have a faint expression and then a faint blue color. you get a higher transformation efficiency with supercoiled plasmids because they are smaller and easier to get into the cells than long and ligated plasmids. If you use plasmid right after ligation, then it is linear and the transformation efficiency is lower and depends on the efficiency of ligation. The top 10 bacteria have a particular characteristic in its genotype that permits its use in transformation with topoplasmid, and it is that they do not contain the laxate gene. It will be provided by the plasmid.
For an efficient ligation and adequate transformation, it is recommendable to add the quantity pointed out by the provider, if it is plasmidic DNA or PCR product, such as equimolar quantities. Another advice could be the usage of adequate cells in which they have to have mutation in genes codifying nucleases and recombinases for reaching an adequate transformation. The competent cell should have the necessary machinery for plasmid replication and transcription. And last but not least, the selection mechanism to indicate the presence of an insert. There are some mechanisms that lead to protein dysfunction, such as the case of beta-galactosidase. Of the usage of reporter gene can be a recommendable choice.